giving rise to. So, just as, I think it was last week, we talked about gluconeogenesis, making glucose anew, the neo bit meaning anew. In other words, from something that wasn't itself a carbohydrate. Here, glucogenic means giving rise, or able to give rise, to glucose. Ketogenic means effectively not able to give rise to glucose, but in fact able to give rise to something called ketone bodies. And this is all very important for animal metabolism. It can be very important for us if our diet is heavily composed of protein, or if we've used up all of our dietary carbohydrate, if we're in starvation, if we're in some form of ultra-marathon type event where every bit of carbohydrate is gone and we have no fat to metabolize, then we'll start breaking down proteins. And depending on the amino acids that we break down, we will either be able to produce glucose or not. And of course that can be very important because the glucose that's then produced can be broken down to make more energy and keep us alive. So what we're talking about here is the ability of the carbon skeletons, the R bits, etc., the non-amino bit of the amino acid, to either give rise to glucose or not. Now, to simplify that a bit more, we're saying, can this amino acid be broken down and lead to gluconeogenesis or not? That's really what we're talking about. We talked about gluconeogenesis. We said that it begins effectively with oxaloacetate, which of course is part of the TCA cycle. But this is where we need to think a little bit carefully. What goes into the TCA cycle? How many carbons are added to the TCA cycle at the beginning? Anybody? Two. Two carbons go into the TCA cycle as acetyl-CoA. It's like acetic acid, it's got two carbons, vinegar. Acetyl-CoA has got two carbons. How many carbons are lost as the TCA cycle goes round? Two. Remember, there's an oxidative decarboxylation of isocitrate, so 6 carbons goes to 5. And then there's another oxidative decarboxylation of isocitrate of alpha-ketoglutarate to form succinyl-CoA. 5 carbons go to 4. So 2 carbons go in, 2 carbons are lost. So the cycle begins with 4 and it ends with 4, doesn't it? Oxaloacetate plus 2, blah, 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 oxaloacetate. So if the carbon skeleton of the amino acid is broken down to acetyl-CoA, can that carbon contribute to gluconeogenesis? The 2 carbons, the amino acid is broken down and 2 carbon atoms are produced as acetyl-CoA. Can they contribute, those 2 carbons, to gluconeogenesis? Yes or no? 2 carbons go in, 2 carbons go out. What's the net value of that? Plus 2 minus 2. 0. So if the carbon as a physical entity enters the cycle in the form of the 2 carbon molecule acetyl-CoA, by the time that cycle has got to oxaloacetate, if you like, those 2 carbons have been lost, haven't they? So you can't make anything with the 2 carbons that went in. You can't make glucose. There's no net excess of oxaloacetate if the carbon enters as acetyl-CoA. Because to get from the start of the cycle back to the beginning, you've got to lose 2 molecules of carbon. So you put them in and you lose them. So any amino acid that produces acetyl-CoA is ketogenic. It is unable to participate, if you like, in gluconeogenesis. In other words, you can't make glucose from breaking it down. And instead, you make these things called ketone bodies, either from acetyl-CoA or a more complicated form of acetyl-CoA or acetoacetyl-CoA. Really doesn't matter, it's too much detail, but just be aware that if the entry point, if the breakdown compound from the amino acid carbon skeleton is acetyl-CoA, then those two carbon atoms effectively are lost going round to oxaloacetate, because oxaloacetate is where we leave the TCA cycle to make glucose through gluconeogenesis. We talked about oxaloacetate needs to go back to phosphoenol pyruvate. Look back at the slides from last week, you'll see that. So I guess another way of looking at it is saying which of the side chains of the amino acids can we break down such that we can add to this right-hand part of the TCA cycle. And they're all highlighted there in pink. These are highlighted in pink as well because they form pyruvate which can enter the TCA cycle through a carboxylation reaction. So those carbons are directly lost as oxaloacetate. But generally speaking, or in addition to that, all of these amino acids add into this part of the cycle where there's 5 carbons. So some of the carbon that's added here is going to be lost because we then go to 4 carbons here, remember? Through the dehydrogenation here, the oxidation step there. But if you add the amino acid as this 4-carbon compound or this 4-carbon compound, if the carbon skeleton ends up as either succinyl-CoA or fumarate, then that carbon here can leave the TCA cycle because it hasn't been lost on the way round. Whereas if it enters over here, there's 2 carbons here, there's acetyl-CoA, goes in with oxaloacetate to form citrate, 6 carbons, 5. One of them's lost. 5 carbons, 4. The other's lost. 2 in, 2 out. Effectively, they were never there. They gave up some energy, but they can't be used for gluconeogenesis. So they're not glucogenic. So this is really important, particularly in animal diets. Okay, now an example of what happens in metabolism, just another example of how well orchestrated, how well organised metabolism is given by this thing, the glucose-alanine cycle. So this is a representation of an arm, a human arm there, and what we've got is a representation that the muscle is working. The muscle, of course, is made up of proteins, and the proteins are made of amino acids. And as we know, as we excessively work a muscle, it starts to sort of break down just a little bit. That's part of the reason why if you don't regularly work muscles and then you've suddenly got to lift a lot of books onto a shelf or something, you'll have sore arms the next day. If you go to the gym after not going there for a while, you're going to know it for a few days afterwards. And part of that is because you've actually started to break down the muscles. You can't sort of completely lose them, but it's part of the process. And what happens in that situation is that the amino acids are deaminated, as we saw before, to form glutamate. So go right back to the early slides. That's happening up here in the muscles. At the same time, of course, the muscle is undergoing glycolysis to produce ATP and producing pyruvate. And the pyruvate, in the absence of sufficient oxygen, will start to accumulate and produce lactate. But an alternative is for the pyruvate to undergo a transamination reaction in the reverse direction to that we saw on the early slides. Instead of forming glutamate, it actually reacts with glutamate, and in those conditions the glutamate loses the amino group to form alpha-ketoglutarate, but more importantly the pyruvate gains it to form alanine. Now at this point the alanine transfers back, obviously not through space, but through the blood system into the liver, which is demonstrated here. The liver then takes the amino group off the alanine and converts that back to pyruvate. So we're simply going pyruvate, alanine in the muscle, get it to the liver, alanine, pyruvate. Reverse the reaction down here. So now form glutamate, go through the whole deamination process, the urea cycle, and the amino group from the breakdown of the protein is excreted in the usual way. But the most important, the interesting bit of this is that within the liver, of course, pyruvate can form glucose through gluconeogenesis. It doesn't happen in the muscles, it happens in the liver. The glucose that's made is then pumped into the blood, because that goes right through the liver, of course, back to the muscle where it's needed. So the muscle gets to do what it's good at, which is be a muscle and contract and use ATP. It needs continuing supply of glucose in order to produce that ATP, that is provided by its breakdown products, pyruvate, taken back to the liver. So again, it's just an example of how beautifully organised metabolism is. We could find similar examples in plants, in their metabolic processes around photosynthesis, photorespiration. Everything is aimed at minimising the inefficiency of the process. And this is a great example of that from amino acid metabolism. Okay, so moving on with great rapidity, which is good. If we just sort of ran everything I've just said about breaking amino acids down in reverse, that's effectively how amino acids are made. So there's nothing complicated. If you grasp how they're broken down, step A, step B, synthesis is effectively step B followed by step A. So as we say at the top here, ammonia is incorporated into biomolecules through the two amino acids, glutamate and glutamine. So we saw how glutamate is made, and then we saw how glutamine is made. So this is that assimilation process. So this is what happens in plants. This is what can happen in our systems if we are remobilizing amino acids that are being broken down from food. Because remember, we don't have a store of amino acids, but we can break proteins down. As cells are continually being replaced, their proteins will be broken down and, if needed, recycled, if you like, to make all the different amino acids. In that case, all the amino groups end up as glutamate, and some of that's then converted to glutamine. So as it says there, the concentrations of those two are where the regulation of the whole process is exerted. We're not going to talk about the regulation in any detail, in fact at all, other than just to mention that it is the levels of those two compounds. Plants, of course, take up nitrogen as nitrate from the soil, and there are a whole series of processes that convert the fixed nitrogen into nitrate that we might have touched on earlier, and I think you've covered also in Foundations of Plant Science. Okay, so effectively, amino acid synthesis is just a case of getting that complicated carbon structure, and there are really complicated pathways for making each of the 19 different side chains. We don't need to worry about those, they just exist. They're all very well characterised. We, as humans, don't have the ability to conduct all 19 of those. Plants do, microbes do. So that's, again, that's the sort of biochemical basis. We can do all of the transamination reactions. So we can stick the carbon skeletons onto the nitrogen groups, but we can't make all of those different carbon skeletons as human beings. So that's why we have the essential amino acids. But once we've got that carbon skeleton, it's really a simple case of adding the amino group onto it using the transaminase reaction that we saw right at the beginning of the lecture. The amino group comes from glutamate. Just as in the breakdown of amino acids, it ended up in glutamate. In the synthesis of amino acid, it comes from glutamate. Again, another example of metabolism being as efficient as it possibly can. Now, just as we saw that the breakdown of the amino acids leads to a range of different compounds which can all enter the TCA cycle, either in such a way as to give rise to glucose through gluconeogenesis or not, as it says here, the TCA cycle intermediates, the amino acid carbon skeletons all come either from glycolysis, the TCA cycle, or another pathway that we didn't have a chance to look at this semester. We decided to drop it because it's just another pathway, a thing called the pentose phosphate pathway. That's yet another way of using glucose. But, analogous to the slide earlier about the breakdown and the production of ketogenic and non-glucogenic amino acids, this shows us the pathways of glycolysis from glucose down to pyruvate, the TCA cycle here with citrate, oxaloacetate, so on and so forth. And it also highlights in dark blue a couple of compounds that come from the pentose, meaning 5-carbon phosphate pathway. And as you can see, those individual pathway steps, be they TCA cycle, be they glycolysis, or be they pentose phosphate pathway, are the basis of the formation of all of the carbon skeletons. So whilst the sort of steps involved are all complicated and specific to each of the 19 amino acids, really what we're talking about is a fairly simple set of common intermediates. Importantly, for what we're talking about today, it's where they're from. They're from glycolysis, they're from the TCA cycle, and they're from this other pathway. That's all. So again, metabolism is being simple, it's being efficient. It's saying glycolysis is not just a way of getting energy, it can also be a way of making amino acids. It can also be a way of storing glucose for future use through glycogen. Another topic we've not had a chance to talk about this semester. But I guess the take-home message is just to sort of think of these metabolic pathways not so much as a hammer, a tool for just one job, but kind of a Swiss Army knife, a multi-tool. It can do all sorts of different things. If you need a screwdriver, it can be a screwdriver. If you need a knife, it can be a knife. If you need to get a Boy Scout out of a horse's hoof, it's got one of those capabilities as well. So some examples of that, just showing the sorts of reactions. We talked about these in the breakdown process, so this is exactly the same in the reverse process. Instead of making glutamate, we're starting with glutamate. That's where the amino group comes from. If the carbon skeleton is pyruvate, then we end up with alpha-ketoglutarate. Remember, as I said at the beginning, it's the same enzyme. It's the aminotransferase enzyme. It's the pyridoxal phosphate. If we looked at the thermodynamics, these reactions under cellular conditions are very close to equilibrium. They'll go in the breakdown or the synthesis path direction, completely dependent upon the concentration of the reactions that are present. So they're close to their equilibrium conditions. Okay, so to summarise, because we can't store nitrogen, because we can't store amino acids, there has to be a balance. Coming in, being used. If it's not going to be used, it needs to be excreted. The point I've tried to make is this sort of central efficiency, if you like, of metabolism, beautifully illustrated by nitrogen metabolism. Using the different pathways that are also involved, as we know, in energy production, in a range of other activities in the cell. The same steps, the same pathways, sometimes the same intermediate compounds are present in a range of different pathways and are used in a number of different ways, depending on what the demands of the cell are. And we saw, as an example of that, the urea cycle, which was the forerunner in Hans Krebs's mind of the TCA cycle. How the urea cycle and the TCA cycle didn't just overlap in one scientist's mind, but actually represent a really good example of how metabolism achieves this incredible efficiency. Because the compounds that are, the intermediates that are involved in both of those pathways there are common intermediates that are involved in both of those pathways. And we also saw with respect to breakdown and synthesis, how the carbon skeletons don't just come out of anywhere, they come from those same central pathways of metabolism. Okay, so there's some good chapters on this in the textbook. And there is a nice little video that brings the urea cycle to life on the Khan Academy. Okay, thank you all very much. Best of luck in your exams. I think I'll see you next week. See you next week. The more civilised time of
11 o'clock.
11 o'clock. Fantastic. Okay, so when are these guys next?
I'm lecturing now.
Straight off?
Yes.
Okay,
yeah,
all right.
Yeah.
Good. Thank you. Yeah, all right, thank you. Can you?
Yes, I can. Yeah.
Proteins and all the nucleotide bases that make up DNA and RNA. So whilst we really haven't talked about nitrogen at all, it is incredibly important. So we're just going to spend this one lecture talking a little bit about the metabolism of compounds, biological compounds that contain nitrogen. So we have our usual outline of the lecture, and we have some learning outcomes that you can refer to. So why are we interested in nitrogen? You'll be aware, of course, that we're breathing this stuff now. 80% of atmospheric gas is nitrogen, but it's not doing anything. We breathe in 80% of nitrogen, we breathe out 80% of nitrogen. From that perspective, we cannot access the nitrogen we need to make amino acids and all of those things just by breathing. That's a pity, but we can't do that. Chemically, it is really unreactive. So N2, nitrogen gas, really doesn't want to do anything. It's pretty damn happy existing as N2. And that's the form that it finds itself in the atmosphere. So our atmosphere is carbon dioxide, CO2, oxygen, O2, and nitrogen, N2. And that is really unreactive. There's a lot of it out there in biological molecules, though, so it is important from that simple perspective alone. In the atmosphere, the nitrogen gas is what we call inorganic. It is nothing to do with life. It is just there. But it is converted into living molecules, into organic compounds, through a series of processes. As says there, biological nitrogen fixation, and that turns the nitrogen gas into ammonium. So we go from N2 to NH4+. And then that ammonium chemical molecules by, as it says there, reactions that we know as nitrification. And then that nitrification leads to the production of amino acids, and those are the steps we'll look at this morning. And then at the end of it all, either when we excrete our excess food, or when we ourselves die, when plants decompose, the protein, the amino acids, the nitrogen compounds are all recycled back through a whole series of microbial processes, ending up back in the atmosphere as nitrogen gas. So you can look up for yourself in any of the textbooks or just online an example of the nitrogen cycle, showing how nitrogen goes from the atmosphere through living compounds and back up the other side, if you like, back into the atmosphere. So the first of those steps is nitrogen fixation. Now this is, if you know, if you're from the ag side of our studies here, you know that nitrogen fixation is a really important process in agriculture because it's a way of taking that inorganic nitrogen and effectively for free, having microorganisms convert that in the soil into the nitrate ultimately that the plants can take up and use for their growth. So nitrogen fixation is a tremendously important biological process in nature and is tremendously important to us in many aspects of agriculture. The reaction up there, nitrogen plus hydrogen, gives 2 molecules of ammonia. It's a hell of a lot more complicated than that. There are people who've dedicated their lives to working on nitrogen fixation. I dedicated 3 years of my life to doing that. Part of my PhD, or my PhD was looking at part of one of the nitrogen fixing reactions. It's a tremendously complex process. But that's sort of this summary of what's happening there. And as you can see, there's no reason why that reaction won't happen. Overall, the products are far more stable than what we started with. So it's a thermodynamically favourable process. The problem is, as I've alluded to before, nitrogen, N2, that molecule is formed by nitrogen forming not 1, not 2, but 3 covalent bonds between the 2 atoms of nitrogen. That makes it really, really stable. So in order to convert nitrogen into what is ultimately ammonia, the activation energy of the reaction is really, really high. Now, if you think back to what we talked about in the lecture about enzymes, where we had the free energy of a reaction here, and we had the progress of the reaction here. You'll recall, I hope, that we said a reaction tends to start with the reacting molecules, and it goes up, and then if it's a favourable reaction, we end up with the products. And this difference in free energy change is the free energy change of the reaction, delta G. Because if that's a negative value, in other words, this is less than that, then it's a favourable reaction, as we see here. And this was the little bit of activation energy that all reactions have. And we talked how an enzymatically catalysed reaction reduces the value of that activation energy. So the reaction is more likely to happen. With the nitrogen fixing reaction, whilst it is thermodynamically favourable, so we can effectively replicate my drawing there for the free energy change of the reaction, the activation energy is kind of way off the scale on my diagram here. It's disappeared off the top of the film. Oh, we're not recording anyway. So the activation energy in this case is enormous because of the stability of the nitrogen-containing compound. So the role of the nitrogenase enzyme in this case is to bring about the reduction in activation energy that is absolutely massive. Only when that happens does the reaction proceed in a meaningful time period. The whole process is driven by the hydrolysis of ATP. As I said, there's a huge amount of, well, we can spend the rest of the year talking about the chemistry of this process. Just in summary, there is a huge amount of energy that has to be expended. So to fix, as the phrase goes, to fix one molecule of nitrogen into 2 molecules of ammonia requires 16 ATP molecules. So it's a very energy intensive process, but nevertheless it is so important because we can't, without this we don't have amino acids and therefore we wouldn't have proteins and we wouldn't have DNA. There would be no life. Then it's just a case of speculating, investing to accumulate sort of thing. And as you can see, on the planet, an awful lot of this is happening. 10 to the power of 11 kilograms, nearly a trillion kilograms of nitrogen gas is fixed in this way into ammonia on the planet per year. So it's a tremendously important process. Once the nitrogen has been fixed, and it says up the top here, ammonium produced by nitrite reductase. You can also write on your notes there by nitrogenase, which is the nitrogen fixing enzyme. So ammonia is also produced by acids, and that's involving this thing nitrite reductase, which we won't talk about anymore today, but both of those enzymes are involved. The phrase we then use instead of talking about fixing the nitrogen is to say that the ammonium is then assimilated. And in a theme that we'll see recur again this morning, of all of the different ways of assimilating, of taking the nitrogen in, it ends up in one particular molecule. In this case, a molecule called glutamine, which, as you may recall, is one of the 20 amino acids. Here's a picture of glutamine here. And we can see one of the important things about glutamine is it's got 2 nitrogens on it. So standard amino acid has, if you remember, the carboxylate group end and the amino group end, a hydrogen and then an R group. Now the R group for glutamine itself, this chain here in pink, contains another nitrogen. So glutamine is a great way of bringing nitrogen into a biological system because it's got 2 of them. So there's a process here, this reaction that's explained here, catalysed by an enzyme called glutamine synthetase. So that involves ATP as well. And the consequence of that is that we end up with fixed nitrogen from the atmosphere being taken through this assimilation process and forming one of the 20 amino acids. And that's the sort of first step in taking inorganic nitrogen and doing something biologically meaningful with it. Now these are the 20 amino acids. As you remember from the very first lecture and hopefully from first year, what makes each of them different from one another is highlighted in the pink shaded boxes here. The 20 different side chains or R groups. Now because there are 20 different amino acids, there are 20 different ways these can be broken down and 20 different ways in which they can be synthesized. So we're going to cover all of those today. But you'll be pleased to know we haven't got 40 slides of different little subtle differences in how it happens because with the exception of one amino acid called lysine, and we can now forget about lysine because we're not going to talk about it again, all of the other 19 amino acids are made in exactly the same way and they're all broken down in exactly the same way. So we haven't got to consider 38 different reactions this morning, we're just going to look at 2 sets of reactions. How they're made and how they're broken down, which is, as you'll see, a lot simpler. But at the end of the day, the complexity of the amino acids is of course reflected in their R groups. I don't need you to learn the chemistry of the different R groups, just to be aware that, of course, going back to our considerations of protein structure and enzyme activity, it is the chemistry of those parts of the molecule there shaded in pink that's responsible for protein function, enzyme activity, and all of those things. Nothing more, nothing less. So this is, as you'll be aware, I hope, an amino acid. And breaking it down, at the end of the day, we've got the amino bit on the outside of that box, and on the inside bit, we've got the bit that contains carbon. 1 and 2 of the R groups have some nitrogen in them as well, but effectively, if we're considering the metabolism of amino acids, we've got 2 things to think about. And this is the same whether we're talking plants, microorganisms, animals, humans, whatever. We've got 2 things to think about. What do we do? What do we do with the N bit, the amino group, and what do we do with the carbon bit? And that's the same consideration whether we're talking about making amino acids or breaking them down. And we'll start with how are amino acids broken down. Plants do this all the time. Plants can make all of their own amino acids. We as humans can't. You'll probably be familiar with the phrase essential amino acids and non-essential amino acids. And you may feel minded to take particular nutritional supplements which are high in certain amino acids, which are often labelled the essential amino acids. The reason they're labelled essential is what? Anyone knows? Take a wild punt.
Because our body can't produce them.
Our bodies can't produce them, so it's essential that we get them from our diets. Plants don't have that problem. Plants can make everything, so can microorganisms, but mammals can't. So, whatever protein there was in the last meal you had is being broken down. The peptide bonds between the amino acids are being hydrolyzed. That's what digestion is all about. And those amino acids in some cases are going to be further broken down, or they're going to be reused immediately to make more protein. Now, unlike fats, which we can store, unlike carbohydrates, which we can store, we don't store amino acids. So most of the protein that you eat is broken down and makes we, basically. So unless you are involved in strenuous activity, or unless you have just completed strenuous activity, once we get to about your age and beyond, we're not growing anymore. So all of the protein we eat, basically just sort of, some of it is broken down for energy. The carbon bits and the nitrogen bits are just excreted. We used to have a whole lecture on sports nutrition and the biochemistry of that. And one of the sad facts there is that most sports nutrition is entirely irrelevant. All you've got to do is just keep drinking and eating a normal diet, and that'll be fine. Anyway, part of that is because we don't store amino acids. We just excrete them if we're not using them. Okay, so the pathways by which amino acids are broken down and made are the same, just in reverse. So it's even simpler again. So we've only got to learn one set of pathways, because it's the same whether you're breaking down the amino acid to its constituent parts or whether you're building up amino acids from their constituent parts. So the first thing that happens in the case of breaking down an amino acid is for the amino group to be transferred from that amino acid. So the NH2 bit is taken away. That is then removed from the molecule it has been added to and excreted. That's all that happens for each of those 19 amino acids that have a common breakdown pathway. And similarly, for each of those 19 amino acids that has a common set of synthesis pathways, what happens is an amino group is brought in, it is added onto a carbon skeleton, and hey presto, you have an amino acid. There's nothing more to it than that. So it's a very simple set of reactions. As it says here, the first one is called amino transfer or transamination. So what trans, of course, means moving across. The amino group is moving across, if we're breaking it from the amino acid, onto an intermediate compound. Then, in the case of breaking down, that amino group is taken off the intermediate compound and excreted in various forms. In the case of building up an amino acid, the amino acid is added to the intermediate form, which is then transferred across to the final amino acid. So that's all pretty much said here. So we can move on and just look at what happens in the transamination reaction. And in another example of a common theme, and metabolism does this a lot. Metabolism has evolved to be as efficient as it possibly can. Doesn't mean we're at the end point of metabolic efficiency, but one of the key tenets behind the way evolution has driven metabolism is to be as efficient as possible. So just as we saw that the fixed nitrogen that comes from nitrogen fixation reactions ends up in the amino acid glutamine, all of the removed amino groups from the breakdown of amino acids end up on this compound, another amino acid called glutamate. So here's the reaction we're talking about at the bottom here. This is where it says L amino acid. That's one of 20 different amino acids, one of 19 different amino acids. It reacts with this compound, alpha-ketoglutarate, a 5-carbon compound, which we're familiar with from the TCA cycle. It's one of the intermediates in that, if you look back at your notes there, but this is not happening in the mitochondria. But it's still the same compound, alpha-ketoglutarate. So whichever of the 19 amino acids we're talking about reacts with alpha-ketoglutarate. The amino group from the amino acid ends up on the alpha-ketoglutarate, which converts it to the amino acid glutamate. The carbon bit that's left behind forms this keto acid. So here we have the carboxylic acid part of our amino acid here on the carboxylate group of the L amino acid, and then we form a keto group here on the second carbon. So we form this keto acid, and that happens for each one of the 19 amino acids. What distinguishes each reaction is a different enzyme. So we have these aminotransferase enzymes, which are specific, as it says there, for the individual amino acids. So proline aminotransferase, glutamate aminotransferase, histidine aminotransferase, etc., etc. There's one for each. And as it says, they have a complicated reaction, which again we don't need to go into, but just as an example, again, the chemistry isn't carried out by the amino acid bit of the enzyme, it's carried out by a cofactor. In this case, something called pyridoxal 5-phosphate. That's where the chemistry of transferring the nitrogen across happens, not in the amino acids. The amino acids give the structure to put the reactive bits in the right place. The same aminotransferase enzyme works in the reverse reaction. Instead of breaking an amino acid down, making an amino acid. So proline aminotransferase is involved in breaking down proline, or if the conditions are around you are set up for protein amino acid synthesis, then proline is made in that way by that same enzyme. Okay, so at that point, if you imagine you've sort of just eaten, I don't know, a burger or something that's got lots of protein in it, all of the different amino acids have been broken down, and all of their amino groups are now sitting in molecules of glutamate. So we've gone from a very complex amino acid profile to a really simple one. And all of those different carbon skeletons of the 19 amino acids are now present as different keto acids. We'll talk about what happens to those in a little while. But all of their amino groups are now sitting as glutamate. And then the glutamate moves into the mitochondria, and then the amino group is lost through a process, as it says up on the board, called oxidative deamination, catalyzed by an enzyme called glutamate dehydrogenase. And we've got a representation of that on this slide, the next one. And we're now in the mitochondria. So we've basically gone back to where we started. Remember, the first step of the transamination involved alpha-ketoglutarate outside the mitochondria. This is happening inside the mitochondria, and alpha-ketoglutarate cannot leave the mitochondria. Remember when we were talking a couple of weeks ago about oxidative phosphorylation, which happens in the mitochondria. Only certain molecules have passage in and out of the mitochondria, and alpha-ketoglutarate is not one of those. So this alpha-ketoglutarate that is produced by the deamination reaction then participates in the TCA cycle, and we'll come back to what the consequences of that are in a little while. At the moment, what we're talking about is what's happening to the nitrogen, which was in an amino group, and has now been knocked off the glutamate by a hydrolysis reaction involving water, and it forms ammonium, NH3, and you'll remember ammonium is a base, so in water it'll attract a proton and become NH4. So now, that's not associated with anything. It's a molecule of ammonia in the cell, which is actually rather dangerous. but it's no longer part of a more complicated biomolecule. So any one of the 19 amino acids, this is how its nitrogen is finally removed if we're breaking down that amino acid. What happens next? This is where our livers come in again, if we're talking in mammalian systems. Fish, microorganisms, birds, reptiles, all do it differently. They have different ways. A fish is very lucky, it's swimming obviously in water, and it can basically just let the ammonia it produces flow away. We can't do that. Birds, of course, produce this incredibly nitrogen-rich guano, bird poo, whatever you want to call it, which of course we have exploited as fertilizer. Why do we use it as fertilizer? Because it's really rich in nitrogen. Because it's what the birds use to get rid of their nitrogen. Mammalian systems use a compound called urea. Now, urea is a very small molecule, which I think that is urea. And urea has a few benefits. It's really small. For a small molecule, it makes it really soluble. Because we don't want this stuff building up inside us and crystallizing. That's not good. So it's also, as you can see, if you add up the atomic weights of the constituent parts, they'll come to 60, of which 8 is nitrogen. Because nitrogen's got an atomic weight of 14. So almost 50% of the weight of this molecule is nitrogen. So this is a fabulous way of getting rid of nitrogen in a very highly soluble form. If you think back to the very first lecture, we talked about Anne Finson's experiment, and we talked about how he took an enzyme called ribonuclease and he dissolved it in a solution of 6 or 8 molar urea. 8 molar urea is 480 grams of urea dissolved in a final volume of 1 litre. That's a huge amount. That's half a bag of sugar dissolved into a litre. Urea is incredibly soluble. In other words, it's not going to crystallise out. So you can get rid of a lot of nitrogen without having to lose a lot of water. Because losing water is not necessarily a healthy thing for an awful lot of animals. You know, we have access to it in bottles wherever we go, but an awful lot of life is not like that. So being able to get rid of as much nitrogen as possible in a very highly soluble form is a really good thing. So the urea is formed through something called the urea cycle that we'll look at in the next few slides. The important thing here is that the ammonia that is made through the deamination process then gets reconverted often back to glutamine or another amino acid called alanine to transport it to the liver, because ammonia floating around on its own is toxic. Now, we talked 2 weeks ago about the citric acid cycle, or the Krebs cycle. The same guy, Hans Krebs, who went on to discover the Krebs cycle, started with this one, the urea cycle. This was the first time a metabolic pathway had ever been considered, conceptualized to be a circular process. Before that, people had understood glycolysis and various things like gluconeogenesis as being linear pathways, but Krebs was the first one to say, you know what, it makes a lot of sense if this is circular. And just as we saw with the TCA cycle, some of the themes that he identified in the urea cycle are then those that enable him to interpret and sort of unravel what was happening with the citric acid cycle. So if we look at this just in outline here, we'll go through the steps in a moment, but just in outline, the urea comes in, the ammonia rather comes in at the bottom, down here, and it reacts with a molecule of carbon dioxide and it forms this thing called carbamoyl phosphate, which is analogous to pyruvate forming acetyl-CoA. That then enters the cycle. It joins up with a compound called ornithine. Now, ornithine has got 5 carbons. Plus 1 makes 6. Now, it's not citric acid, in this case it's citrulline. The 6-carbon citrulline leaves the mitochondria, and then in the cytoplasm it reacts with another amino acid, and of course being an amino acid, that will have an amino group on it, and it forms this That's got 6 carbons, aspartate's got 4, so it forms this 10-carbon compound called argininosuccinate, which, most importantly, has got 2 amino groups on it. So 2 molecules of nitrogen are now sitting on this one compound, but it's big, it's got 10 carbons. It then loses 4 of those to produce fumarate. You may remember fumarate is part of that TCA cycle. And we'll look at how these 2 cycles not only came from the genius of one person's thinking, but actually metabolically are linked as well. We'll look at that in a moment. So we lose, from our 10-carbon compound, we lose 4 as fumarate. That gets us back to 6, an amino acid called arginine, which has got 2 amino groups, very close to one another on this side chain. That is then hydrolyzed using water, and this little bit chops off, urea, just as I drew it on the board. So small compound, carbon, oxygen, 4 hydrogens, and 2 molecules of nitrogen. So we've then gone from 6 up here, down to -1, down to 5. And then that leads back into the mitochondria, and we start the cycle again. So Krebs worked this out as being the way in which the urea cycle works, and then he applied that same principle to the TCA cycle. He got a Nobel Prize. So that's all you've got to do. If you want to go to Stockholm, just do something like that and you'll get a Nobel Prize. So this is some detail. I'm not going to go into too much of this detail, but again, you can come back and have a look at this. But this is basically what's happening. The ammonia that's produced from the deamination reaction reacts with carbon dioxide or bicarbonate in water. Again, some ATP is required, and it produces this molecule, carbamoyl phosphate. The carbamoyl phosphate then reacts with ornithine to produce citrulline. That then leaves the mitochondria, so by this point we're up to a 6-carbon compound. That then reacts with a 4-carbon amino acid, aspartate, to produce a 10-carbon intermediate, argininosuccinate. And you can see, I'm skipping over a lot of the detail, but you can see, if you look back, things like ATP, there's a lot of ATP involved in this process as well. So a lot of us get rid of ammonia, but it's such a toxic compound, you've got to do that, otherwise you're going to die. The 10-carbon compound argininosuccinate then breaks up to form arginine, which has got 6 carbons, and fumarate, which has got 4. And then the arginine, which has got the 6 carbons but has also got the 2 amino groups on it, is hydrolyzed to produce the urea and the ornithine. And the ornithine then starts the cycle again. These are the names of the enzymes that are involved. So, a number of those compounds, as I said, are common to both the urea cycle and the TCA cycle, which enables the cell that this is happening in to be more efficient. Because it can use those compounds, it can shuttle the compounds between one or the other reaction, depending on what's happening at any one time. The same compound can serve 2 or more purposes. And so we can see some detail here of where the aspartate comes from a transamination reaction, which is the sort of reaction we talked about right at the beginning of the slide, beginning of the presentation this morning. The alpha-ketoglutarate is involved in making aspartate. Again, we know that's part of the TCA cycle. Fumarate, we know that that's part of the TCA cycle. Yeah. This is all diagrammatically represented here by various textbooks and pictures online that show this. They sometimes refer to it as Krebs's tricycle or Krebs's bicycle or that sort of idea. The idea is to represent the 2 cycles, the citric acid cycle, shown here within the mitochondria, and the urea cycle, shown at the top right, partly within the mitochondria and partly on the outside, in the cytosol. This diagram, there's a little bit here called the malate aspartate shuttle. You can just put a line through that. We're not talking about it today, it just makes things more complicated. But this was the best diagram I could steal from a textbook. And what this demonstrates, again, is that interaction between the 2 cycles. So we have the TCA cycle here producing oxaloacetate, and then we can transaminate that to form aspartate. The aspartate can leave the mitochondria, and as we saw then in the urea cycle, it reacts with citrulline to form the intermediate, which then breaks down, producing another TCA cycle intermediate, fumarate, which is converted back into malate, and the malate goes back into the mitochondria. So again, without worrying too much about the detail, just get the sense that these pathways don't happen in isolation of one another, either physically within the cell or in terms of what the cell is doing. And this is the same whether it's a plant, an animal, or a microbe cell. That depending on the metabolic requirements of the cell, the same compound can serve a number of different fates. Okay, so that's what happens to the nitrogen. So it starts off in protein as amino acids, gets broken down, and effectively the urea is then passed out in urine in mammalian systems. That's got rid of that excess nitrogen because we can't store it, we don't store amino acids, and the amino groups that are taken from amino acids are toxic, so we get rid of them. That leaves all of those complicated bits of carbon, the 19 R groups and the other backbone of each amino acid. And there are 19 different ways of breaking those down. But we don't, again, need to discuss all 19 of them individually. We can group them in this way. We can look at the compounds that we're familiar with. We've heard of all of these. Acetyl-CoA, alpha-ketoglutarate, succinyl-CoA, fumarate, and oxalo acetate. We've heard of all of these because they're all part of the TCA cycle. Now, depending on which of these compounds the amino acid side chain breaks down into, and it's generally either one or the other, but some of the more complicated side chains have enough carbon in them to form one or more of these compounds. Depending on which one of those 5 compounds is formed, will determine a very important part of the property of the amino acid in terms of its metabolic value, which I hope is illustrated on the next slide. Yes, it is. Good. So we can classify amino acids as either being glucogenic or ketogenic. Now, genic